students, and welcome to another session for environmental biotechnology. We will start this class soon at 3:05, and let's wait for some more people to join and we can start this class. Yeah, I think it's time to start this class. So I welcome you all to another week, week 3 of environmental biotechnology. This is a live TA session in which we will be solving some questions related to all the lectures that you have encountered in week 3 of environmental biotechnology. So sorry for the inconvenience that I was not able to take this session on Saturday, but I made sure that I had prior informed the NPTEL team to send you the link and the updated timings, since I was traveling on Saturday 3 to 5 p.m. Right? So I hope you have all gone through your lecture week 3 by Professor Pinaki sir, the course Environmental Biotechnology. So we will solve some example questions from the assignment which I have designed for revising the topics that you read in lecture week 3. This is a general set of instructions for all students who are participating in this class as well as who will be watching this recording later. This is a recorded class in which we will be solving some problems, 10 or 11 problems clearing doubts of students in this course, and the slides and presentation that I am going to present in this class will also be shared to the NPTEL and will be made available to the students. This video recording will be uploaded on YouTube channel and the link which will be provided to you by the NPTEL authorities. But I highly encourage everyone to attend these live sessions, to interact with each other, to interact with me, and get a hands-on experience on how to solve these assignments and clear your doubts through me. This will be an interactive session, but I will advise everyone to please turn off your mic and video chat unless absolutely necessary. You can interact with me via the chat box. I'll be following it regularly. And with that much of an introduction, let's start today's session. So we will start today's sessions by solving the doubt which one of these students had with the final question of the lecture week 2. So let me first share my screen and just give you a minute. I can share my screen. I'm sorry. Okay, I hope everyone can hear me properly, loud and clear, and can see my slides moving. I'm trying to move my slides. If yes, then let me know in the chat box. And for everyone who is participating in this course, please interact and try to answer your questions. It's okay if you do not know the answers. We will go through each question very briefly. We'll spend time on it. We'll understand the options, the concepts, and understand why is the correct answer the chosen correct answer, right? So that we also revise the topics as we go along solving these questions. Okay, thanks for your response. So once again, I welcome you all to another session for live TA session for this course, Environmental Biotechnology. This course is taken by Professor Pinaki sir at IIT Kharagpur, and my name is Ashutosh, and I'm a PMRF scholar at IIT Kanpur, the teaching assistant for this course. There is another teaching assistant for this course associated, so you can attend both these lectures or any of these live sessions that you want to. And this will be the format of this presentation. I have prepared 10 to 11 questions for all of us. This is a 2 hour long session in which we will be dealing with such multiple choice questions, of which only single option is the correct answer. Single or multiple options correct. And I will post these questions. I will ask you what do you think is the correct answer. You post your responses in the chat box, and let's take a minute or so to analyze all the responses, to collect many responses as possible. I see that the class today is very thin, very less number of people, but it's okay. Many people will be watching this recording later in the future, so we will give ample time and then discuss these answers as we move forward. Okay, so starting off with the revision question from lecture week 2. This was the question that we got stuck at, we got confused. I'll read it aloud for everyone. Many biogeochemical reactions are carried out under anoxic conditions. Which one of the following is not a suitable explanation for this trend? So in lecture week 2 we were dealing with biogeochemical cycle, and the question here tells us that such reactions are carried out under anoxic conditions, and which of these is not a suitable explanation for this trend? And here are the options. Biogeochemical cycling involves redox reactions. The directionality of these reactions are changed by ambient oxidation reduction potential. Option B, biogeochemical cycling reactions are harmed by light exposure and only occur in aphotic environments. Aphotic means where light does not reach, which are also usually anoxic. Option C, biogeochemical cycling involves reactions catalyzed by metalloenzymes which are susceptible to oxidation, which makes them non-functional. So here option C talks about the metalloenzymes which get non-functional in an oxidative environment. Biological reduction of elements other than oxygen tends to yield less energy. Hence, most organisms prefer to utilize oxygen wherever it is available. And option E, biogeochemical cycling involves ancient microorganisms that evolved before the great oxidation event and therefore are naturally susceptible to oxidative stress. So these were the 5 options, and we were wondering which option is the correct answer. I will make a quick revelation that option D is the correct answer. This is the final correct answer. Biological reduction of elements other than oxygen tends to yield less energy. Hence, most organisms prefer to utilize oxygen wherever it is available. So this is the correct answer to this question. This explanation is not a suitable one for many biogeochemical reactions happening under anoxic conditions. Now, if we look at all these options, option A, biogeochemical cycling involves redox reaction, which is very true, and the directionality of these reactions can be changed by reduction oxidation potential. So it is counterintuitive that redox reactions are the main constituent of these biogeochemical cycling, and directionality of these reactions, which when I mean is whether there is more oxidation or more reduction in a reaction which will lead from reactants to the products, because from whichever direction these redox reactions move will determine the directionality and also the change in biogeochemical cycling. For example, carbon and nitrogen biogeochemical cycles, direction can shift based on environmental oxidation potential, often favoring anoxic conditions when oxygen is scarce. So, what happens when an oxygen is scarce in an anoxic conditions? When oxygen is scarce, oxidation reactions directions reduces and shifting the direction of the biogeochemical cycles to the anoxic conditions. B. Biogeochemical cycling reactions are harmed by light exposure. So we know that in a pond ecosystem, mostly the aphotic zone, which is the zone where there is less or no light, is also coinciding with the anoxic zone. So this is also a suitable explanation because many processes like sulfate reduction often occurs in deep sediments which are anoxic and aphotic, and light can inhibit many microbial activities like sulfate reduction. Option C, where biogeochemical cycling involves reactions or enzymes catalyzed by metals which are susceptible to oxidation. So the enzymes which are in coordination with metals, the metals oxidation stage is what matters to the function of the enzyme. So if the oxidation state of the metal is changed, then it dislodges from the enzyme and renders the enzyme inactive. That is why metalloenzymes, for example for nitrogen fixation, need anoxic conditions to maintain a proper oxidation state of the metal which is coordinated as function to the enzyme, such as manganese in nitrogenase. Now option D is not the suitable explanation because other than oxygen there are electron acceptors like sulfur and sulfate and nitrate in anoxic conditions which are used by microorganisms as alternative electron acceptors. We all know of bacteria which can use iron, sulfur and nitrate as electron acceptors. Of course, oxygen is the most energy yielding electron acceptor, but in absence of oxygen, which is in anoxic conditions, these these compounds are also acting as the electron acceptors. Thus, the preference of oxygen doesn't explain the prevalence of anoxic biogeochemical reaction. It doesn't really mean that only when oxygen is present and only when oxygen is present there can be biogeochemical cycling. And option E, where biogeochemical cycling involves ancient microorganisms, because the example of great oxidation events which occurred around 2.5 billion years ago, such before this event the only microorganisms that used to thrive on the earth are anaerobic microorganisms which are sensitive to oxygen, favoring anoxic conditions for their role in the biogeochemical cycle. That is why option E is also a correct explanation, A, B, C is also a correct explanation, but option D is not a correct example. So here is a representative diagram of all the different kind of processes undergoing in a biogeochemical cycle in a pond ecosystem. The oxic zone, which is also enriched in as a photic zone, where there is oxygen and enrichment of the sunlight, is where oxygen is fixed, carbon is fixed by bacteria, methane. Methane oxidation, blue bacteria, methane oxidizing bacteria oxidize methane to CO2 in the presence of oxygen. This primarily occurs in the oxic zone near the surface. And organic matter production, methane oxidizing bacteria generate methane organic matter which sinks to the anoxic zone where iron reduction and iron cycling happens. So this is an example of methane oxidizing bacteria and iron reducing bacteria which are which are there in the carbon cycle as a biogeochemical cycle. So option D is the correct answer, which is not a suitable explanation for why many reactions are carried out in anoxic conditions, because other than oxygen there are many other electron acceptors like sulfate and nitrate which happen to prefer which happen to localize in organisms in the anoxic zone where oxygen is not available. I hope the doubt from the previous lecture is clear to the students. Option D is the correct answer. Is it clear to the students? Just let me know in the chat box that not only oxygen but many other elements can act as electron acceptors for this reaction. Very good. Thank you. Moving on to the first question of this session. lecture week 3. Thermophilic bacteria from hot geysers of Yellowstone National Park, USA, was the source of the valuable enzyme dash. I'll repeat the question once again. A thermophilic bacteria from hot geysers of Yellowstone National Park, USA, was the source of the valuable enzyme dash. Option A, pepsin. Option B, tyrosine. Option C, chymosin. Option D, 529 DNA polymerase. Option E, Taq DNA polymerase. This should be an easy question to answer. Thermophilic bacteria, very famous, isolated from Yellowstone National Park hot geysers. There is an enzyme which is a valuable enzyme in today's molecular biology experiments, isolated from this bacteria. What is the name of this enzyme? Let us take a minute and collect some answers in the chat box, and then I'll come back to this. Sorry. Yeah, then let's come back to this question. Yeah, I think it's time to reveal the correct answer. I can see that Saida has answered option E. Okay. And since there are less number of students in the class today, let us try to look at what is the correct answer. Correct answer to this question is option E, Taq DNA polymerase. Congratulations, Sahida. Sahida has answered correctly. Taq DNA polymerase is a valuable enzyme which was isolated from Thermus aquaticus from the Yellowstone National Park in USA. Okay. So Taq polymerase, why is this enzyme so crucial? Taq polymerase is an enzyme used in polymerase chain reaction, which is commonly known as PCR. PCR is one of those techniques, pioneering techniques, which revolutionized molecular biology forever, right? And this bacteria, this enzyme was isolated from its species Thermus aquaticus, a bacteria which is an extremophile bacteria found in hot geysers, where there is hot springs inside the Yellowstone National Park, in the geothermal areas. And this enzyme, this polymerase present inside the bacteria, which is an RNA polymerase, DNA dependent DNA polymerase, is a heat tolerant enzyme allowed for the development of PCR, a technique that revolutionized molecular biology. So Taq polymerase is a DNA dependent DNA polymerase. It carries out DNA replication, which led to the boom of PCR technology, in which you can amplify any DNA of your interest through polymerase chain reaction. So isolation of this enzyme revolutionized molecular biology from the Yellowstone National Park. Now, what is the key feature of this Taq polymerase? Key feature of this polymerase is to function at high temperatures, particularly around 72 degrees Celsius, which is optimal temperature for DNA synthesis during PCR. So this is, since DNA is a double-stranded helix, is a very heat-stable molecule, you need enzymes which catalyze this reaction at higher temperatures, unless facilitated, so that there is ample denaturation of the DNA, opening of the double helix, and then amplification or polymerization by this enzyme. So here is a schematic showing how the polymerase, Taq polymerase, works on the DNA. There is a 5 prime primer and extension. There is a primer to which Taq DNA polymerase at higher temperature can add on new nucleotides, amplifying the target sequence. In PCR application, DNA is heated to separate its strands, and then the Taq polymerase is used to synthesize new DNA strands by adding nucleotides to a primer. So there needs to be an existing DNA molecule which has hybridized onto the template, and then Taq polymerase can polymerize or extend the primer in complementarity to the base sequence of the template at higher temperatures such that DNA strands stay separated. Key properties of the Taq polymerase includes 5 prime to 3 prime polymerase activity and 5 prime to 3 prime exonuclease activity. These are the two key features of the Taq polymerase. It has 5 prime to 3 prime polymerase activity. It adds onto the tail of the existing primer in 5 prime to 3 prime direction, and it also has exonuclease activity, or cutting off a wrongly pieced DNA and allowing the correct sequence to sit on as DNA is replicated. But it lacks 3 prime to 5 prime exonuclease activity, meaning it does not have proofreading ability. This property results in PCR product with a 3 prime DA overhang. So these are some of the applications in which Taq polymerase isolated from Thermus aquaticus has been used for. That this polymerase has 5 prime to 3 prime exonuclease activity and 5 prime to 3 prime polymerase activity. I hope this is clear to the students. This was an easy question, first question for this week, lecture week 3. If yes, then let's move forward in the discussion. Okay, thank you, sir. Saida says, yes, sir. You're welcome, Saida. Okay, moving on to the next question of this session. Question number 2. Which one of the following options correctly describe the superbugs? If you remember what are superbugs, you will be able to answer these questions. Which one of the following options correctly describe superbugs? Option A, insects with enhanced biodegradation potential. Option B, microorganisms with enhanced biodegradation potential. Option C, genetically engineered insects with enhanced biodegradation potential. Option D, genetically engineered microorganisms with enhanced biodegradation potential. Option E, isolated protoplast with enhanced biodegradation potential. Which of these can correctly describe the superbugs? 5 options, only one is correct. Remember the definition of a superbug and what is its application, and you will be able to answer this question correctly. Which one of the following options correctly describe the superbugs? Let us wait for a minute for everyone who is watching this video as well as those who have joined the live class. Please post your comments in the chat box. What you think is the correct answer to this question? Saida has answered option D. Okay, Saida. We'll wait for some time and then I'll reveal the correct answer. Okay, I think it's time to reveal what is the correct answer to question number 2. Which one of the following options correctly describe the superbugs? Correct answer is option D. Option D is the correct answer. Genetically engineered microorganisms with enhanced biodegradation potential. These are what describes the superbugs very correctly. So once again, Saida has answered the correct answer. Genetically engineered microorganisms with enhanced biodegradation potential are nothing but superbugs. So superbugs for biodegradation are genetically engineered or naturally occurring organisms with enhanced capability to break down pollutants. They are called superbugs because they are able to do something which none of us have ever thought about, degrading pollution and helping us in bioremediation and biodegradation. These superbugs often utilize specific enzymes or metabolic pathways to degrade substances like hydrocarbons, plastics, or chlorinated compounds. So, all of these substances are hazardous and harmful to our nature, but when released in the environment, such superbugs can be able to fight these pollutants with specific enzymes and metabolic pathways degrading hydrocarbons, plastic, and chlorinated compounds. So on my right is a design of a superbug from characteristics with the characteristic of 4 different bugs. So let us suppose there is strain A bacteria with CAM plasmid. Now what is a CAM plasmid? CAM plasmid contains an enzyme which can degrade camphor. Now camphor is a aromatic hydrocarbon, long chain hydrocarbon. So a bacteria has CAM plasmid, enzymes in the bacteria can degrade camphor. Then strain B is a bacteria which has oct plasmid or OCT plasmid which can degrade octane. Octane is another harmful non-polar long chain aliphatic carbon compound hydrocarbon. A bacteria which can degrade octane is an oct plasmid, while strain C has xyl plasmid or XYL plasmid, xylol, in which there is an enzyme that can degrade xylol, and NaH plasmid degrades naphthalene. So we have here 4 different strains of bacteria degrading variety of long chain hydrocarbon compounds as pollutants. Now what we would do is hybridization or sexual conjugation of these 2 bacteria, DNA hybridization and sexual conjugation of strain A and strain B bacteria to create a strain E bacteria which harnesses both the power of the CAM plasmid as well as the oct plasmid. The genes coding for the enzymes for degradation of camphor and octane came into strain E due to sexual reproduction between these 2 bacteria or conjugation. Hybridization, the same hybridization process produces strain F in which there is XYL and NaH plasmid, and another step of hybridization between strain E and F, and of course there is antibiotic selection to select for certain superbugs which have all the 4 enzyme coding regions. Thus this superbug strain G can degrade camphor, octane, xylol and naphthalene all at once. So this can be used as a multifaceted biodegradation microbe useful in creating a pollutant free environment with long chain fatty acid hydrocarbons. So this is how a superbug is genetically engineered or from the naturally occurring microorganisms and can incorporate different powers and potential of the enzymes and metabolic pathways to degrade the pollutant of interest. For example, why an insect is not called a superbug? There was an option which said that insects which are genetically engineered enhanced biodegradation potential. The insects which have genetically engineered and have bioremediation potential are known as bioremediation agents. For example, wax worms. This is a wax worm example, a moth, in which the moth larva feeds on plastic. And I have mentioned an example here, species Galleria melonella, known as the greater wax moth. They can degrade plastics of certain types such as polyethylene tetraphthalate through action of the saliva and enzymes and the gut microbes that they harbor. So these are insects which are not genetically modified but naturally occurring, like wax moths. They have special enzymes in the saliva which can degrade plastic, and also the microbiota that are present in their gut, they also have the capacity of digesting this plasmid. So such agents are commonly known as bioremediation agents. But superbugs are genetically engineered microbes from naturally occurring organisms or artificial selection, where a single bug can harbor multiple powers or multiple enzymes degrading multiple harmful pollutants in the environment, like the one which we saw in the previous example. So in this life cycle of the adult moth of the waxworm, degradation initiates with the saliva enzymes when the larva is feeding on the plastic, and it continues with microorganisms of the gut enzymes. And when the larva disposes the residue, it completes the life cycle. This frass contains all the degraded products of the plastic, and it completes its life cycle when it releases or disposes the residue and turns back into the adult moth. So such agents are called bioremediation agents, while such microbes which when combined due to DNA hybridization and sexual reproduction of bacteria produces superbugs which can degrade camphor, octane, xylol and naphthalene. So Saida was here with the correct answer, and is it clear to the students? What are superbugs? These are microbes which are genetically engineered to do an enhanced biodegradation for multiple variety of pollutants.
टेक्निकल इंजीनियरिंग की खास प्रोटीन प्रोटीन जैसे जेनेटिक इंजीनियरिंग केवल डीएनए की मुझे समझ आ गया
Okay, someone has a question. Dheeraj has a question. Dheeraj, do you have a question?
डीएनए को कट करना और उसे सही डीएनए जोड़ना यही जेनेटिक इंजीनियरिंग का काम है ना सर यही
Yes,
कहना चाहता
yes,
था
yes.
मैं
Okay. Thank you, Dheeraj. You can, so I will advise again to all students, please turn off your mic and video chat. You can communicate in the chat box if possible. आप प्लीज़ अपना माइक एंड वीडियो ऑफ कर सकते हैं. चैट बॉक्स में आप अपना कमेंट्स पोस्ट कर सकते हैं, which I'll be following for smooth conduction of this class. And the mode of the mode of communication would be English, since this is a global class, and recordings will be available for all other students to refer in the future. Okay, moving on to the next question, question number 3. Question number 3: biological nutrient removal is the process of dashed. Biological nutrient removal. Option A: recovering nutrients from food waste. Option B: recovering nutrients from sewage. Option C: completely removing nitrogen and phosphorus from wastewater. Option D: completely removing carbon and potassium from wastewater. Option E: developing a microbial community of aerobic, anoxic, and anaerobic bacteria for the use. Which of these would explain biological nutrient removal? Let us wait for a minute and and collect some answers. All the students participating in this live TA session, please post your answers in the chat box. Whatever you think is the correct answer. Biological nutrient removal. Okay, I think it's time for me to reveal the correct answer. Let us check the comment box for whatever answers we have collected. Only Saida has answered option C. Any more answers? Any more students who want to answer this question? Biological nutrient removal is the process of recovering nutrients from food waste, recovering nutrients from sewage, completely removing nitrogen and phosphorus, completely removing carbon and potassium, or developing a microbial community of aerobic, anoxic, and anaerobic bacteria. Which of these options is the correct one? Let me know in the chat. Okay, since there are no more answers in the chat box, let me reveal the correct answer to this question. Correct answer is option C. Completely removing nitrogen and phosphorus from wastewater is referred to as biological nutrient removal. So Saida has again answered the correct one. Well done. So what is biological nutrient removal process? Biological nutrient removal is a wastewater treatment process that uses microbes to remove nutrients, primarily nitrogen and phosphorus, to prevent eutrophication in receiving water bodies. Okay, so this is very simple to understand. Biological nutrient removal. So you want to remove all the nutrient biologically from the wastewater before releasing it into the recipient water body or in the river. So it is a wastewater treatment strategy, which is the biological nitrogen removal. Not just nitrogen, other materials like nitrogen and phosphorus are removed maximally from the wastewater before release into the recipient water body to prevent eutrophication. And eutrophication happens due to enrichment of the water bodies in nitrogen and phosphorus, which leads to algal bloom. So to prevent eutrophication, this wastewater is treated for biological nutrient removal. Now, two steps for biological nutrient removal. Nitrogen removal, the first step. Microbes convert ammonia into nitrite and then nitrate. So the dissolved ammonia in the wastewater is converted into nitrite and nitrate under nitrification with aerobic conditions. In an anoxic zone, other bacteria use this nitrate as an oxygen source, converting it into nitrogen gas, which is released to the atmosphere. So we have nitrification in aeration tank, and then denitrification to release nitrogen gas back into the oxygen into the atmosphere. Phosphorus removal. Certain bacteria like phosphate accumulating organisms or PAOs take up phosphorus in excess under alternating aerobic and anaerobic conditions, storing it as polyphosphate. This excess phosphorus is then removed when biomass is wasted. So phosphorus removal happens by bacteria like polyphosphate accumulating bacteria, and which take up enormous amounts of phosphorus in and store it as polyphosphate. So phosphate is removed from the organic matter or in the wastewater as polyphosphate, and this excess phosphorus is removed when biomass is wasted. So BNR is widely used in sewage treatment plants, enhancing the water quality by reducing the nutrient nitrogen and phosphate content that can cause algal blooms and oxygen depletion in ecosystems. So before releasing any organic matter or any wastewater to the water body, it undergoes BNR to reduce the levels of dissolved nitrogen and phosphorus and then put it back into the water stream. So this is the schematic of a flow by flow biological nutrient removal chamber. So the effluent from the primary treatment, once the primary treatment is done, where bigger particles are settled down and the sludge sediment is settled down, it comes into anaerobic chamber in which dissolved and organic matter where the bacteria is growing in fermentation and creates ammonia. This ammonia goes into the anoxic zone, the next chamber, in which it is converted into nitrates. Now nitrates is then released by other bacteria, denitrifying bacteria. Nitrates is again acted upon, and nitrogen is released back as nitrogen gas, while the effluent is then treated into an oxygen rich phosphorus polyphosphate containing organisms chamber, in which, in the presence of oxygen, the phosphorus is stored as polyphosphate, and then this outflow, this effluent is then fed into the secondary clarifier, in which the activated sludge is returned back to the anaerobic chamber to enhance this process, and the clarified water is then fed into the water bodies. The removal of excess sludge and organic phosphate from the sedimentation secondary clarifier, and the activated sludge returns, while the remaining sludge goes for further treatment. So this is a whole schematic of biological nutrient removal, in which the levels of nitrogen and phosphorus in the wastewater organic matter is decreased via nitrification and denitrification. I hope it is clear to the students, and Saida has answered correctly. If it's clear, then give me a thumbs up in the chat box and we can move ahead. Biological nutrient removal. Okay, cool. Looking at the fourth question of this session, which of the following is not a possible advantage of modern technologies like membrane bioreactor over conventional wastewater treatment systems? This is a question about MBR or membrane bioreactor systems. The question is about which of the following is not a possible advantage of modern technologies like membrane bioreactor over conventional wastewater treatment systems? Option A, MBR require low space requirement. Option B, MBR have low surface area. Option C, MBR are more efficient. Option D, MBR can use H2 to reduce oxidized contaminants. Option E, MBR give better effluent quality. Which of the following is not a possible advantage of membrane bioreactor? Let us take a minute and collect some answers in the chat box, and I will reveal the correct answer in a minute or so. If you remember what is a membrane bioreactor and how does it look like, you will be able to answer this question. Okay, I think that's enough time for this question. And let's see what is the correct answer. Let me first check the comment box for some answers. Saida is the only one answering option B. And I have to tell you that option B is the correct answer. MBR have low surface area. This is the correct answer to this question. Low surface area of the MBR having a low surface area is not a possible advantage of membrane bioreactor. Now let us understand what is a membrane bioreactor and how does it look like. A membrane bioreactor is a wastewater treatment technology that combines membrane separation process like microfiltration with ultrafiltration with a biological treatment process, typically an activated sludge system. So we just encountered activated sludge system in the biological nutrient removal system. So this, when coupled to a physical microfiltration membrane or an ultrafiltration process, becomes a membrane bioreactor. On my right is a diagram showing the structure of a basic structure of a membrane bioreactor, where the wastewater coming in uh, is under aeration tank, where there is an aeration bioreactor in presence of oxygen. The organic matter is broken down, and then there is a membrane filtration setup in which aeration membrane system, in which this activated water after treatment from the aeration bioreactor is being fed into the membrane system, and treated water can be reused, while excess sludge is then put back as activated sludge into the aeration bioreactor. The only difference is that a membrane bioreactor also does physical filtration like micro or ultrafiltration coupled with the aeration biological treatment process. Membrane bioreactors offer several advantages in wastewater systems. High quality effluent. MBRs produce clearer water with low turbidity and pathogen levels due to fine filtration of the membranes. This was obvious since there is a membrane now filtering the effluent after aeration biological process treatment system. This creates water which is more cleaner, which is higher quality before release into the recipient water body. Small footprint. They combine biological treatment with membrane filtration, reducing space needed compared to conventional systems. So they are not very large in size and they occupy less space and doing similar similar or better facilities than a conventional wastewater treatment system. Improved solids removal. Membranes effectively retain biomass and solids, enhancing treatment efficiency. Of course, it will only allow liquid to pass through as a membrane, and it will retain much of the biomass in the form of activated sludge, which can then be used to to increase the efficiency of wastewater treatment. Flexible operation. MBRs can handle variable loads and are suitable for retrofitting existing plants. So it can handle a decent amount of load from the wastewater area. And reduced sludge production. The process can minimize excess sludge, lowering disposal costs. This can also reduce the excess sludge production, which consists of oxidized organic matter along with bacterial biofilms. The process can minimize excess sludge, lowering disposal costs. MBR is high quality, producing high quality effluent. It has a small footprint, combining the filtration along with biological wastewater treatment systems. It retains solid and active biomass and can minimize excess sludge, lowering the disposal cost. So on my left is a organic matter rich wastewater which is treated upon by microbes, and here it shows the property of solids removal, in which membranes can effectively retain biomass and solids, enhancing treatment efficiency, only leaving clean water to leave the membranes and sent into the water bodies, recipient water bodies. I hope it is clear to the students. the membrane bioreactors, and what advantage do they confer in comparison with traditional biological reactors. I hope it is clear to the students. Just let me know in the chat. Okay, thank you for the confirmation. Moving on to the next question. Question number 5. Which of the following statements about microbial ecosystems is false? Which of the following statements about microbial ecosystems is false? Option A. Microbial ecosystems can also exist inside macroorganisms like humans. Option B. Microbial communities behave like a single multicellular organism. Option C. Biogeochemical parameters drive microbial ecosystems at macro scale. And option D. Single cells cannot be ecological units. Option E. Meta communities operate at centimeter to meter ranges. I will advise everyone to read through the question very thoroughly and just let me know in the chat box what do you think is the correct answer to this question. Which one of the following statements about microbial ecosystem is false or not true? You first have to remember what is a microbial ecosystems, who are the players, what kind of players or participants in this ecosystem, and which of the following statements is false about microbial ecosystems. Okay, I think it's enough time for me to reveal the correct answer to this question. Which of the following statements about microbial ecosystem is false? I think Saida has answered option B. Microbial communities behave like a single multicellular organism. Okay. Which of the statement is false? And the correct answer is option D. Single cells cannot be ecological units. This is not a true statement. This is a false statement. Single cells indeed are considered as ecological units. I'm sorry, Saida, but option B is not the correct statement. Correct answer. It is a false statement. It is a true statement. Microbial communities behave like a single multicellular organism is a true statement. Now, what is a microbial ecosystem? A microbial ecosystem refers to the complex community of microorganisms like bacteria, fungus, and viruses interacting with each other and their environment. So here is an example of a mangrove ecosystem in which there is root exudes from the mangrove ecosystem. There are bacteria which can fix the atmospheric nitrogen, and then denitrifying bacteria which can reduce the nitrates present in the soil via denitrification to release atmospheric nitrogen. There are sulfates reducing and iron oxidizing bacteria. The root exudates and the nutrient absorption profile of the phosphate solubilization is carried out by one group of bacteria. pH degradation, pH stands for polyaromatic hydrocarbons, which are pollutants, and metal absorption as well, while some other bacteria are important in decomposing the organic matter. So all of these bacteria as well as viruses comprise the microbial ecosystem, and these interactions can be crucial for processes like nutrient cycling, decomposition, and even health of larger organisms including humans. So such microbial communities or microbial ecosystems should cooperate with each other to carry out complex processes like nutrient cycling, decomposition, and health of larger organisms including human. Single cells in a microbial system can be considered an ecological unit. This was the correct answer. In microbial ecology, individual cells such as bacteria or fungi can function as independent units capable of performing any key ecological roles. So, individual cells can function independent units to perform key ecological roles such as nutrient cycling like nitrogen fixation, degrading pollutants as we saw in the previous image, polyaromatic hydrocarbons, or living in symbiosis with other organisms like the rhizobium symbiosis in the leguminous root nodules. Each cell can respond to its surroundings, reproduce, and contribute to ecosystem balance, making it a fundamental ecological unit within a larger microbial community. So, single cells are also known as the fundamental unit of ecological community. They can act and respond to surroundings, reproduce, and contribute to the ecosystem's balance. So, therefore, analyzing the options again, option A, the human microbiome is a clear example of microbial ecosystem existing in that existing inside larger organisms, which is absolutely true. Microbial communities behave like a single multicellular organism. True in many cases because microbes show coordinated behavior and division of labor, which makes them functionally similar to a multicellular organism. So, in a multicellular organism, we observe things like division of labor, distinction of the functions that are being carried out by different types of cells and the players inside a community, and so does happen in a microbial ecosystem with different kinds of microbes. They show coordinated behavior and division of labor. Option C, biogeochemical parameters drive microbial ecosystems at macro scale. This is also a true statement because the availability of nutrient depends on the nutrient cycles, pH, temperature, and oxygen availability all influence microbial community on a larger scale. And option E, which says meta communities operate at centimeter to meter ranges. This is also true because in ecology, meta communities describe multiple interacting communities over spatial scales from centimeters to meters. This is the correct range for meta communities. So the only false statement was single cells cannot be ecological unit. This is the only false statement, which is the correct answer to this question. I hope everyone agrees with me. Option D was the correct answer to question number 5. Okay, I think it is clear to the students. If you have any doubt or you want me to go through this question again, I can teach it back to you. Let us move to the next question, question number 6. In microbial waste treatment systems, reduced organic matter from waste acts as dust. In a microbial waste treatment system, reduced organic matter act as dash. Option A, source of electrons and carbon. Option B, sink of electrons and source of carbon. Option C, sink of electron and carbon. Option D, source of electrons, sink of carbon. Option E, sink of electrons and carbon dioxide. The question is about reduced organic matter present in microbial waste treatment systems. What can it act as? Does it act as a source of electron and carbon, a sink of electron, source of carbon, or both sink of an electron and carbon, source of electrons, sink of carbon, or sink of electrons and CO2? Let us wait for some time and collect some answers. I will reveal the correct answers in a minute or so. Okay, I think it's time for me to reveal the correct answer to this question. In a microbial waste treatment system, reduced organic matter from the waste act as option A, the source of electrons and carbon in this system. So option A is the correct answer, and Saida has given again the correct answer. Option A is the right answer. Well done, Saida. So what happens in a microbial waste treatment system and what is reduced organic matter known for? In a microbial waste treatment system, reduced organic matter acts as a source of electron and carbon because it consists of carbohydrates and proteins, and it is the key source of electrons for microbial respiration, such as in anaerobic digestion, and provides carbon for growth and energy production. That is the only source, the reduced organic matter, which is a starting point of any waste treatment system, is the source for electrons as well as organic matter, which I mean is carbon, provides electrons for respiration, such as anaerobic respiration, and carbon for growth and energy production inside the ecosystem. Sink of electrons and source of carbon. A sink implies that acceptance of electrons such as oxygen or nitrate, which is not the reduced organic matter. Therefore, it is not a sink of electron rather than a donor of electrons or source of electron. Oxygen and nitrates and phosphates are the final electron acceptor or which act as sink of electrons. But it is definitely a source of carbon, but since B has sink of electrons, it is not a correct statement. Sink of electron and carbon, again this is false because reduced organic matter does not act like sink for electrons or carbon. It releases electrons and provides carbon to the microbes as their food. Source of electrons correct, but sink of carbon against false, and sink of electrons and CO2. This is also false because reduced organic matter is generates CO2 during aerobic degradation and donates electron. So this is the last step in which it donates the electron to oxygen and nitrogen as the electron acceptor and releases CO2 through aerobic respiration. So as an excerpt taken from the lecture itself by Professor Pinaki sir, reduced organic matter donates the electron to produce biomass, to produce carbon dioxide, methane and hydrogen as the byproducts. This is the carbon source for microorganisms which metabolize and oxidized this reduced organic matter by accepting electrons to form biomass to release carbon dioxide, methane and hydrogen. I hope it is clear to the students that reduced organic matter in a wastewater treatment system acts as electron and carbon source, not the sink. The sink of electrons is oxygen and carbon is released as carbon dioxide back into the atmosphere. It acts as a food for aerobic respiratory bacteria. I hope it is clear to the students. We are moving fast ahead since there are less people and we are also getting correct answers from very few students here. So we are moving relatively fast. We have finished the sixth question in our series, and if there is no doubt until now, then we will move on to the next question. Okay, I would take that as a yes because there is no doubt in the chat box or no one raised a hand. Let's move on to the next question. Question number 7. Cultivation independent approaches like metagenome analysis is a modern alternative to pure culture study. Which of these is an incorrect comparison of the two techniques? I will read the question again. Cultivation independent approaches like metagenome analysis is a modern alternative to pure culture study. Which of these is an incorrect comparison of the two techniques? Option A, pure culture has only one microbial strain. Option B, metagenome has multiple microbial strains. Option C, more than 1% of microorganisms can be isolated as pure culture. Option D, 99% of all microorganisms can be obtained in metagenome. Option E, DNA of all microorganisms can be obtained in metagenome. Which of these correctly fits the question's answer? Which is an incorrect comparison between two techniques, the metagenomic technique, culture independent, and pure culture technique. Let us take a let us take a minute and come back to the answers in the chat box. Okay, I think it is time to reveal what is the correct answer to this question. Cultivation independent approaches like metagenome analysis is a modern alternative to pure culture study. Which of these is an incorrect comparison of the two techniques? Option A, pure culture has only one microbial strain. Option B, metagenome has multiple microbial strains. Option C, greater than 1% of all microorganisms can be isolated as pure culture. Option D, 99% of all microorganisms can be obtained in metagenome. And E, DNA of all microorganisms can be obtained in metagenome. And of course, the correct answer, which is the incorrect comparison, is option E. DNA of all microorganisms can be obtained in metagenome. This is an incorrect statement. Rest all the statements have some degree of truth to it. Now let us dissect what is a pure culture and what is a cultivation independent metagenomic analysis. A pure culture is a laboratory grown population of a single microbial species, allowing for detailed study of its characteristic in isolation. Once you know which species to study, you can culture it as a pure culture on your bacterial plate or a laboratory homegrown population to study the single species in higher detail. On the other hand, metagenomics analyzes genetic material directly from the environmental sample, encompassing all microorganisms present without the need for culturing. That is why metagenomics is also known as culture independent technique or analysis of the microorganisms. Whereas pure culture, you need to culture the organism to study it in detail. Metagenomics directly analyzes the genetic material from the environmental samples. Now, there is another observation which is very generally made, which is known as the Great Plate Count anomaly. This refers to the observation that number of microbes directly observed from the environmental sample is significantly higher than the number of colonies that can be grown in a laboratory setting using traditional culture methods. Point to be noted very importantly, which shows a stark difference between traditional culture, pure culture method, and metagenomics. The amount of microbes or different amount types of microbes we observe under a microscope, very less or very few of them can be grown in a laboratory setting using traditional culture methods. Not every bacteria can be grown in a laboratory setting. Therefore, great plate count anomaly says that number of microorganisms is significantly higher than the number of colonies that can be grown in a laboratory setting. Now, going through an option by option approach, pure culture has only one microbial strain. By definition, it is true. It is only a culture of one microorganism. Metagenome has multiple microbial strains as it works on the sample directly taken from the environment. So it has metagenome sequences capturing the genetic material from all different microorganisms present in a sample. Greater than 1% of microorganisms can be isolated as pure culture, roughly around 1%, less than 1% organisms. Looking at the significance t-test values of 0.005%, the Great Plain Count anomaly says that less than 1% of microbes can be cultured. So greater than 1% is correct in the sense that only a small fraction can be isolated. So see, only a small fraction out of all the organisms can be isolated and grown in laboratory condition. So C might be false, but some cases it holds truth. 99% of all microbes can be obtained in metagenome. This is also a true statement because it can access most of the microbial DNA, including uncultivable species which cannot grow as pure culture. While DNA of all microbes is an overstatement, It cannot be obtained in metagenome. While metagenomics can capture DNA from most microbes, it's not truly all. Some may be missed due to extraction biases, sequencing limitation, and low abundance. So, low abundance and depth, low depth sequences are a problem in metagenomics, in which it cannot capture the 100% genomic signature, but definitely significantly more than what a pure culture can capture. It promises around 99% of all microorganisms that can be captured in a metagenomic analysis. So, option E is the incorrect statement and the correct answer to option 7. We understood what is the Great Plate anomaly, we understood what is the pure culture, and how is it different from metagenomics, and why metagenomics cannot claim to identify 100% of all the microorganisms present in the environment. I hope it is clear to the students. If yes, just let me know in the chat box and we can move ahead with the next question. Very good. Thank you for the response. Moving ahead to, okay, so there is a table that I have mentioned here which compares metagenomics and culturomics before moving ahead to the next question. On the left side, we have the definition, process, limitations of the methods, benefits of these methods, and future outcomes. Metagenomics is a technology that describes microbial composition using high throughput sequencing method of 16S rRNA sequencing. Culturomics technology describes microbial composition using high throughput culture method. This uses 16S rRNA as a technique. This uses MALDI-TOF MS, which is mass spectrometry for identification. Limitations of metagenomics: no strain is provided for further identification, does not recognize minority populations, depth issues. Yeah, this is the reason why 1%, 99% of all the microbes can be identified, but not 100%. Only can find eubacteria, no information generated on enzymatic capabilities. Culturomics may not find some non-culturable microorganisms as in pure culture. Pure culture is a subset of culturomics. Increase workload to perform the testing. So you have, this is not a high throughput method, while metagenomics is a high throughput method. Detects non-culturable microorganisms, but culturomics detects minority populations. Culturomics addresses the only disadvantage that metagenomics approach has. It can successfully identify 200 bacterial species per sample, while culturomics only a few. Okay, moving on to the eighth question. Which one of the following options is true for a comparison on aerobic and anaerobic microbial process for wastewater treatment? You have to identify the correct statement for a wastewater treatment involving aerobic and anaerobic chambers both. Option A, aerobic microorganisms grow faster than anaerobic microorganisms grow faster than aerobic microorganisms. Option B, slow growing aerobic microorganisms are preferred in waste treatment. Option C, aerobic metabolism yields less energy than anaerobic metabolism. Option D, anaerobic fermentative growth is the fastest in treatment technology. Option E, nitrifiers and methanogens are aerobic. These are the 5 statements. Which one of this is true for a comparison on aerobic and anaerobic microbial processes of wastewater treatment? Let us take some minutes and get back to the comment section to check how many answers have been written in. I would encourage every participant to answer these questions, to interact with me via the chat box. We are in question 8. 2 or 3 more questions and we will close up this session. Okay, I think a minute has already passed, and let us check for the answers in the comment box. Rashid has answered option A, and there is no more answer. Saida is answering option E. Okay, anaerobic microorganisms grow faster than aerobic. Nitrifiers and methanogens are aerobic. Okay, now let us see what is the correct answer to this question. A comparison of aerobic and anaerobic microbial process for wastewater treatment. Correct answer is option B. Slow growing aerobic microorganisms preferred in waste treatment. Now, if you remember the lectures that were taken by Professor Pinaki, he gave ample time on explaining why a slow growing rate is preferred by microbial engineers in wastewater treatment. Let us understand the concept in a bit detail with excerpts from his own lecture series. The main aim of wastewater treatment is to retain a slow growing biomass because most of the environmental biotechnology uses specific growth rates which are slower than the microbiology laboratory. Whatever growth rate is there in a laboratory, the actual real life wastewater treatment plants use a much slower rate. Because why do engineers use such slow growth rates? Because of two reasons. Because methanogens and nitrifiers, which are also a part of the waste water treatment facilities, these bacteria cannot grow quickly, especially nitrifiers, which grow at a rate of 0.07 per day, whereas aerobic heterotrophs are fast growing species such as 0.25 per day. So, because of them, methanogens and nitrifiers, eventually these guys also have to produce the biomass slowly for nitrification and methane generation. And also, processes that exploit slow growing bacteria slows the specific growth rate down to less than 0.07 per day. Reliability and stability demand. Slow processes create a reliable and stable organic matter demand. So the features of aerobic versus anaerobic microorganisms in a wastewater treatment plant. The main electron acceptor is oxygen, while anaerobic it is nitrate, sulfate or carbon dioxide. Energy yield is high for aerobic microorganisms. So which rules out option A and D, because aerobic microorganisms have higher energy, therefore they grow faster. So anaerobic microorganism cannot grow faster than aerobic because aerobic produces higher energy, and anaerobic fermentative growth is fastest. Again, this is wrong because aerobic microorganisms produce higher energy, and their energy production as well as increases the rate of growth of their species. It is faster than anaerobic fermentative growth, which only produces 2 ATP, 38 ATP in comparison to 2 ATP. Growth rate, aerobic is generally fast, example aerobic heterotrophs, while anaerobic have very slow reproduction rates such as methanogens. Methanogens are archaeas which can take as long as 3 to 10 days in doubling time. And here are some of the examples, and the role in wastewater treatment is very important. Aerobic microorganisms help in carbon removal, formation of activated sludge, nitrification and phosphorus removal, while anaerobic microorganisms help in sludge stabilization, methanogenesis and denitrification. So methanogens are anaerobic while nitrification or nitrifiers are aerobic. Aeration required, energy intensive for growth of these aerobic microbes, while no aeration required, energy saving mode in anaerobic microorganisms. Higher removal efficiency and faster treatment for aerobic microorganisms, lower sludge production and methane recovery by anaerobic microorganisms. So use of anaerobic microorganisms and methanogens slows the startup, produces a slow growing biomass. Therefore, such processes are often chose engineers often choose such slow specific growth rates for cultivating both the systems of aerobic heterotrophs, which are initially fast but then they choose slow type of aerobic heterotrophs to match the speed of nitrifiers and methanogens, which are anaerobic in nature. Looking at the other options, nitrifiers are usually aerobic while methanogens are usually anaerobic. Nitrification, the process performed by nitrifiers, need oxygen. Methanogenesis is carried out in absence of oxygen. So this rules out the other option. Nitrifiers and methanogens are not both aerobic. Most of the nitrifiers are aerobic, but methanogens are anaerobic. Aerobic metabolism yields much more energy because oxygen is the most efficient terminal electron acceptor. Example, glucose oxidation yields 38 ATP, while only 2 to 4 ATP in fermentation. And fermenters are not the fastest in wastewater system. Fermentation can be rapid in lab conditions, but in wastewater treatment, aerobic systems achieve faster organic degradation. So hence it is clear that slow growing aerobic microorganisms are preferred because of two reasons: to maintain a stable and reliable demand, and because the bacteria used, methanogens and nitrifiers, are slow growing bacteria. Hence the engineers use a slower rate than what they use in the laboratory. Okay, is it clear? It is clearly mentioned in Professor Pinaki Sar's lecture series as well. He has spent a lot of time in explaining why slow growth rates are preferred in wastewater treatments. You can go back and revise your lectures in lecture week 3 and you will be able to understand this concept very clearly. Okay, thanks for the confirmation, Rashid. Moving ahead with question number 9. New molecular tools for analysis of microbial communities should meet four primary criteria. Which one of the following is not one of those criteria? New molecular tools for identifying microbial communities maintain four primary criteria. Which one of these is not one of those criteria? Option A, they should be inexpensive and simple to perform. Option B, they should reveal structure as well as function. Option C, they should focus on eukaryotes and viruses as well. Option D, methods must be fast and high throughput. Option E, they should be able to separate lead actors from small contributors. Which one of the following is not one of those criteria? Let us take some time and post our comments in the chat box, and I will reveal the correct answer in a minute or so. Okay, I think that's enough time for collecting the answers. Let me check the chat box. Rashid has answered option E. Ashwini says B and Saida says C. Okay. Lot of different opinions from students. I hope you have read the question correctly because the question is very tricky. Which one of the following is not one of those four criterias which should be followed while designing new molecular tools of analysis for microbial communities? Correct answer to this question, question number 9, is option A. They should be inexpensive and simple to perform. This is not one of the primary four criterias to make new tools for molecular analysis of communities, while all other three are. I'm surprised. Option B should reveal structure and function. Option C focus on eukaryotes and viruses, and option D must be fast and high throughput, and separate lead actors from small contributors. All of these are important criteria. Now let us recollect, revise what Professor Pinaki has mentioned in his lectures. This is an excerpt showing that new molecular tools for analyzing microbial communities can be evaluated based on four key criteria. It should be fast, it should be high throughput, should represent diverse microbe community, and also provide structure function information. These criteria can help assess how effectively a tool can differentiate between microbial species, how many samples it can analyze, and the depth of information it provides about the microbial community. Correct? Understood? So, in the metagenomic approach, where we take environmental sample and subject it to 16S rRNA sequencing, it can tell us what are the microorganisms that are there present in the environmental sample. So, it can provide information about the diverse microbial community. And let me inform you, it is not just bacteria, but rather eukaryotes and viruses and archaea may also be present in the environmental sample, which should be importantly analyzed by the new molecular tools. Our approach should not be limited to the bacteria as well as exploring viruses as well. So option C is not one of the criteria. Option C is one of the criteria, it's not the answer. What are they doing? So this question tells us about what is the structure and function of this community. Structure, when I mean say structure, how is one group of microbes connected to another via a community, and what are the enzymes which actually perform the molecular functions which is important for the microbial community to do in the environmental sample, and how are they doing it, what are the metabolic pathways, what are the genes that are getting activated in the microbial community. Is it methane oxidizing rich bacterial community, is it a nitrification community, then what are the enzymes that are present in such communities. So new tools should keep all these structures in mind and create an approach which can tell us what different kinds of microorganisms they have. It should be fast, it should be high throughput, so that consumes less of a sample and gives us more information about different microbial species, what they are doing, what is the phenotypic potential of the community, and how they are doing it, what are the enzymes that are getting coded in the community as part of the analysis. So this is integrated into metagenomics approach and various other new molecular tools which are being based on the metagenomics approach to be developed for better analyzing microbial communities. Now, to integrate the omics approaches, most molecular interrogation has been directed towards DNA focused on selected genes aimed at high throughput genomics, total metagenomic DNA of a community or a sample, analysis of 16S rRNA and other functional marker genes, high throughput genomics, which tells us the enormous potential for expanding capability in environmental genomics. Not just the transcriptomics, but incorporating all the possible omics and together as a multi-omic approach like proteomics to understand what is the protein signature of the community, and metabolomics, which Professor Pinaki sir has mentioned, that how a community utilizes metabolic pathway for consumption and conversion of metabolites and cycling them throughout the community. Such a signature from the communities can be utilized in metagenomic approach in designing newer tools which can integrate all the three omics approach: DNA focused transcriptomics, which understands the mRNA and DNA level information; proteomics, which understands the enzyme level information; and metabolomics, which understands the interaction of pathway and metabolic utilization of the microbial communities. And this calls for investment of resources and funding and finances, thus making option A the correct answer. Inexpensive is not one of these criteria because these analyses mostly require a lot of money, and development of better techniques, integration of the omics approach, and techniques which provide structure and function relationship of the microbial communities, along with studying diverse bacterial, eukaryotic, archaeal, and viral communities, will require a lot of funding and will not be inexpensive. Therefore, option A is the correct answer. So, Professor Pinaki sir has mentioned that new tools are calling in for investment of such resources into develop multi-omics approaches for most molecular interrogation to understand the composition of a microbial community and utilize it for environmental uses. I hope this is clear. The four pillars of new molecular tools for analyzing communities: it should be fast, should be high throughput, can analyze a diverse microbe community other than bacteria, and also provide structure and function information on what is the actual phenotypic potential of the community, through which enzymes and metabolic pathways is the community able to achieve such function. I hope it is clear to the students. If yes, then we will move on to the final question of this session. Question number 10. Great. Moving on to the final question, question number 10. When organic waste is disposed in a water body, the resident microorganisms degrade it. Which of the following is not likely a result? Understand the question properly. When organic waste is disposed in a water body, resident microbes will degrade it. What is not likely a result? Which is not a result of the following? Option A. Provided the waste is biodegradable, the microorganism shall completely detoxify it. Option B. Depending on waste composition, algal growth may be enhanced. Option C. Introduction of allochthonous carbon is a natural process which is excessively increased by this waste. Option D. Biodegradation depletes organic carbon content and releases methane and CO2. Option E. Biodegradation enhances oxygen content, driving growth of aquatic fauna. Which of these statements is not likely a result of organic matter being degraded by microorganisms? Let us take some time and collect some answers in the chat box. Final question of this session. I would encourage all the participants present. Please let me know what you think is the correct answer. I will get back to it in a minute. Okay, I think it's time to reveal the correct answer to this question. Only Rashid has answered option E. Ashwini is saying option A. Okay, let us see what is the correct answer to this question. When organic waste is disposed in a water body and microbes degrade it, which of the following is not likely a result? Option E is the correct answer. Biodegradation enhances the oxygen content, driving growth of aquatic fauna. This is the correct answer. This is not likely a result when organic waste is put inside the water body. Rashid has answered correctly. Congratulations. But I'm sorry, Ashwini. Option A is not the correct answer. Let us understand what happens when biodegradation happens of the organic matter option by option. If the waste is biodegradable, all microbes shall completely detoxify it. This is true because if biodegradable, if the organic matter is biodegradable and not toxic, microbes can mineralize it into carbon dioxide. All the microbes will reduce the organic matter and oxidize the organic matter and produce CO2 out of it and methane. But in reality, some compounds may leave residual toxicity. But provided the condition that the waste is biodegradable, everything will be converted into carbon dioxide. Depending on waste composition, algal growth may be enhanced, which is true. Nutrient rich wastes, such as those rich in nitrates and phosphorus, can trigger eutrophication leading to algal blooms. That is why we see eutrophication in ponds and lakes, in which there is a thick layer of algae growing in the surface of the ponds. Introduction of allochthonous carbon is a natural process which is excessively increased by this waste. So, allochthonous meaning coming from external sources like fallen leaves or detritus, dead organic matter from outside of the aquatic system or sewage. Organic waste increases this load excessively. The allochthonous matter will of increase the organic load of the waste. Biodegradation depletes organic carbon content and releases CH4 and CO2, which is true. This is coinciding with option A. It will deplete organic carbon content and oxidize them into methane and CO2. In aerobic zone there is CO2 release, and in anaerobic zones at sediments there is methane production by methanogens in the anoxic region. The only false statement here is option E. When biodegradation consumes dissolved oxygen, which is known as BOD or biochemical oxygen demand, it causes hypoxia or anoxia, which harms fish and other aquatic life. So this is what happens, which is opposite of what mentioned in the option. Biodegradation consumes oxygen, creating biochemical oxygen demand, depleting the oxygen present inside the water and killing marine life in process. So what is biochemical oxygen demand? BOD is the amount of dissolved oxygen that microorganisms need to break down organic materials in water, because consumption of organic materials needs oxygen in the form of respiration, aerobic respiration, in which CO2 is released. Healthy water contains a balance of dissolved oxygen, microorganisms and organic materials. When water contains excess organic materials, if the green dots are more, microorganisms begins to break them down, and as they break them down by respiration, they use up dissolved oxygen, depleting oxygen levels and harm the aquatic life. This is the biochemical oxygen demand, which is the result of organic waste being disposed in a water body, and therefore biodegradation enhances the oxygen content is false. It actually depletes the oxygen content, driving the death of aquatic flora and fauna. So this is not likely a result of wastewater being disposed in a water body, as resident microorganisms degrade it. Therefore, option E is the correct answer. I hope it is clear to everyone that why option E is the correct answer, which is not the true statement, which says biodegradation will enhance growth of aquatic fauna. Okay, cool. So with this, we have come to the end of this session. I will stop sharing my screen once again, and So yeah, thank you for being with me. With this, we have completed some example questions corresponding to lecture week 3, where Professor Pinakisar has completed various range of topics from wastewater treatment, from developing new tools for metagenomics community, from explaining uh, how aerobic and anaerobic respiration helps us in treating the wastewater, and many other topics. How in industrial settings that microbial, we all encountered the questions in how industrial settings and in real platforms, what is the growth rate, which is very less than what is found in the laboratory. So all these topics have been covered. Please go through your lectures and let us meet this week Saturday 3 to 5 p.m. to discuss week 4 assignment questions, example assignment questions corresponding to lecture week 4. And I hope you liked the class, the format of the class. If you have any suggestions, any doubt for me, then please let me know. Otherwise, I will just end this call and end this class. Thank you for joining. If you have no more doubts, I will try to close this class and end this call. Thank you for joining and see you on Saturday. The recording of this class will be available on YouTube, and the presentation slides that I have made for the example questions will also be made available to you. Thank you. I'll end this call now.